one student who's speaking into the door, please turn off the light. It's okay. So have you revised the first lecture at home?
A little bit.
Is it difficult for you?
No, it's easy.
No, it's easy, right? And the first lectures we studied about cells, all the things surrounding the cells, when this were discovered, the cells theories, and also we studied about the domain of life, how all the organism models can divide in with in the X group, we have a big group is the domains, and small subgroup we have the kingdoms, and inside a kingdom we have more smaller group we call the subgroup of the kingdoms. Okay. So from lesson today to the end of the lecture, we study deep inside the cells, the molecule that's compared to cell, the structure of the cells, how everything work in the cells, and our cells, cell culture and application of the cells. And now we move to the lesson number 2, the chemical component of cells. And this part is more, I think it's more
Difficult.
No, it's not really difficult. It's a little bit related to chemistry, and in biology we call them the biochemistry. So I think so many knowledge in this lecture is you have experience in your high school. So I hope you guys are comfortable and enjoy learning it. Some knowledge you have already learned. And the lecture today contains 3 parts. In the first part, we study about the chemical bonds, how all the molecules or the chemical inside a cell can connect to each other. And the second part is we study about the smaller molecule in the cells, and the last part is about the macromolecule in the cell. We move to the first part about the chemical bonds. Forget about my school. So, who can tell me what's made up all the matter in the world? The smallest units. Elements. Elements or so made up all of the matters is made of combination of the elements. And you remember the pictures? I showed you one graph in the lesson number 1. This is one graph about how big a cell is, and you can see that the smallest thing represented in that picture is the atoms. What is the size of 0.2 nanometers? Atoms, right? So the element is makes up all the matter in the world, and the smallest particles of an element, actually if you remember they are small at the same, that's the smallest particle that still retained its distinct chemical properties, we call them an atoms. So have you ever seen a periodic table of elements? Sure? In high school? You saw it so many times, right? So the atoms, we can call them the building block of every element. And every compound, molecules or elements are come across to be made of an atom. And the characteristics of substance. other than the pure elements, including the matter parameter. Are all the matters in the world depend on which atom they contain and the way these atoms link together into group we call the molecules. So to understanding the living organisms, it's important to know the chemical bond that's how these kinds of atoms together. So why the molecules are formed. So that's why we need to study in the first part about the chemical bond. So here's a simpler graph of these atoms. So an atom, they consist of nucleus in the centers, and surrounding we have the electron cloud. So here is the schemes represent an atom of carbon and an atom of hydrogens. So like everything else, you can see that the atom have a few different things, floatings around inside them. And we have here the electrons, they have the negatively charged, protons, positive charged, and neutrons, they are neutral. So, the different atoms, the same elements can have different masses. You can see that if they have too many or too few neutrons in the nucleus, and the nucleus may be not stable, and they will be disintegrated by radioactive decays. You can see here, for example, in the here with the atoms are carbons. So, for example, atom carbons may have the atomic number 6. So they have 6 protons, but they could have either 6 neutrons or how many neutrons do you know? 6 or? So we can carbon 12 and also carbons 14. So it means that they could have 6 neutrons or 8 neutrons, so it depends. So the former case 6 neutron is familiar in chemistry, but for the 8 neutron, they are there's a lot of common light element used in biology. So the scientists can use the carbon 14s to see the pathway of one chemical. To see the pathways of our chemicals or substance or anything else that whirls around inside a cell or inside of an organism, they use carbon 14s. Then, if one kind of atom, they have the different types of neutrons they are different isotopes of that atoms. So you can tell me what the difference between the atom, element, and the molecules. We talk about molecules, we talk a lot about the elements, and we talk a lot about the atoms. So what difference from them? They are the building blocks of matter. So what difference between atom and elements? In high school, I think you experience a lot. So sometimes we get confused because that's element they are. Next, please.
An element is many different atoms with the same number and a proton.
Yes, I agree. We have sometimes you get confused because elements actually they are atoms, and the element they are just a group of all the same kinds of atoms, right? And what are the molecules?
Molecules are created by many different atoms and connected to each other.
Yes, the molecules is forms when so many atoms combines. It's called the same atoms or different atoms. So unlike elements, molecule can be made of same and different elements. So we have, for an example of molecules, we have hydrogens, oxygens, carbon dioxide, etc. Right? So here is the graph show the distribution of elements in the Earth's crust and how they different from the elements inside organism bodies. So you can see in the Earth's crust, you have a cyclic behind numbers you have the oxygens, cyclic aluminum, calcium, magnesium, potassium. So many other elements, but inside inside the living organism, the high levels, we have the hydrogens, oxygens, carbon, nitrogen. So this represents how the elements inside the living organism different from the element in the S-curve. So what could determine how an atom can interact with its others? They have a nucleus with neutrons and protons, and surrounding them you can see electron clouds. So the outermost electrons of one atom can determine how they interact. So if they have incomplete filled electron cells, it makes them less stable, and they tend to interact with other atoms, so can gain a lot enough electron to achieve and complete the outermost cells. You can see that the hydrogens here is only one electron cell. They need one more to get stable, so they can combine with others hydrogen to share the electron in the outermost shell, and they have we have hydrogens in H2 in the gaseous form. So all the elements commonly found in the living organism, they have the outermost shell not completely filled with the electron you can see in the red part. And because they don't have the filled electrons in the outermost shell, they can participate in chemical reaction with other atoms. For the inert gas, we have a helium, neons, and argons, because they have a complete outermost shell, so you cannot find them in the living organism because they cannot interact with other chemicals. So they gains a lot of electron to have the complete outermost shells leading to two types of chemical bonds. So if two atoms gains a lot of electrons, we have the covalent bonds, and for the others, when one gives and one gains a lot, we have the ionic bond. So if two atoms sharing one pairs of electrons, we have the covalent bonds, and we go to discover these types of bond in the next slide. The first type of bond, we have a covalent bonds. So there are strong forces between the atoms form covalent bonds when two atoms they share one pair of electrons or multiple pairs of electrons. So you can see here one carbon plus 4 hydrogens. We have the metans gases. So you can see here they can share the pairs of electrons. So here is 4 pair of electrons having shared. So the connections, the bonding formed between the carbons and hydrogen. Here we have the covalent bonds. So the numbers, the electric structure of an atom determine the number and geometry of covalent bond is handmade, and you can see the numbers of covalent bonds formed by carbon atom in these tables. So hydrogens form only one covalent bond, right? For oxygens, they can form only 2 covalent bonds, but however they have 2 other additional pairs of electrons that they can participate in non-covalent interaction. And when sulfurs here can make 2, 4, or 6. So in 4 maybe you have H2S, hydrosulfate. Maybe 6 you have sulfuric acid. Yes, for nitrogens, it can make NH3, you have the ammonia, and you have these 4 covalent bonds in the ammonium ions, and it's 4 plus, right? For potassiums, 5 covalent bonds, for example, in the acids. Phosphorus, you have the H3PO4, right? So all your experience in high school, so I just remind you. And for in the covalent bonds, the electrons may share equal or unequal. These typical examples you have in the extent of an atom ability to attract electron is called the electron or negativities or we can call them the affinities to the electrons. So you can see here in the oxygens, because they form of the 2 same atoms, so they have the equal covalent bonds. However, for the water, for the waters, because these that's the oxygens, they have the stronger attract to the electron than the hydrogen, so they have more negative here in oxygens, and because the electron is more attracted to the oxygens, so that's why in hydrogens you have more positive here. So we call the depose moments in the water molecules. So I give you some examples of nonpolars. Nonpolars in the bondings share equal between 2 atoms, and in case you have the carbon-carbon or the carbon and hydrogen bond. Polars, when the atoms have different electron negativities, so they have unequal sharings of the electron. And one example here, you have the bonding between the oxygen and the hydrogens. And for the next part, we study for the reds, we have the non-covalent bonds, and the most important thing, we have the ionic interactions. Ionic bonds can form by gains a lot of electrons, while covalent bond we have shared, right? And the ionic bond results from the attraction of positive charged ions, we call it cations, and for the negative charge of an ion, then we have the anion. So you can see the examples when one atom here's two atoms shares electron, we have a covalent bonds, and two atoms that they can transfer electron from this atom to the others to make the complete cell in the both atoms. We have the ionic bonds. That's the difference between two kinds of bonds. And one example of the ionic bond you have in the sodium chloride. So in the sodium atom, they have one more in their outer shells, so it's made sodium is unstable. So they want to give one electron to another atom, and luckily the chlorides they have only 7 in the outer shell, so they tend to receive one more electron. So if the best combine between the sodiums and the chlorides, we have sodium chloride compound, and the interaction, the bonding between the sodium and chlorides, we have the ionic bond. The next bond types, we have the hydrogen bonds. Hydrogen bonds here is put here. So interactions of the partial positive charge between hydrogen atoms. I can put here one, and you can see one here. Positive charged hydrogens in a molecule decompose. So with unpaired electron from other atom in the A here, unpaired electron we have a two here. Or either in the same and the next hydrogen bond here. So here's hydrogen atoms. You see that they form a covalent bond with only one other atoms. It's in the D here. So DX in the DX covalent bonds may form an additional weak association with one acceptor atom A here. So the pairings non-bonding pairs electrons variables. And one favorite example is the hydrogen bonds you see in the water. So in the previous slide on top, the water is decomposed, right? The common bond between the hydrogens and oxygens, we call the unequal bonds. So oxygen is stronger electronegativity, so they attract more the electron to them, so they have the negative charge, whereas the hydrogen, they have the more positive charge. So hydrogen bonds can form between two water molecules by the weak interaction between the hydrogens and the one water molecule to the oxygens in the second water molecule. So you know that's because this is really important, because the water account for 70% of the human body. So the most intercellular reaction occur in aqueous environment. So the hydrogen bonds in the waters make them make these characteristics of them can build the living organism on Earth. The next types of bonds it's just introduced, but we don't go so far as we have a one-to-one interactions. That's when any two atoms approach each other is close and they can create a weak non-specific attractive, and we call the one-to-one interaction. And the last weak bond hydrophobic interaction. Here in an aqueous environment, you can see that there's two hydrophobic molecules. They driven together by hydrophobic effect, and thereby they reduce the extent of their direct contact with water molecules. So for low entropies to the higher entropies, by two hydrophobic molecules interact with each other. Right? Hydrophobic là kỵ nước ạ. Okay, it's hydrophobic interaction. Two hydrophobic molecules where they close together, they they are formed a hydrophobic effect. They stay close together to make them stronger because they do hate water. They don't want water anymore. So they stand closer. We have another weak bond called hydrophobic effect. So we have here the bond strengths. This is, you can image just covalent bonds are much stronger and more stable than non-covalent interactions. And they are very stable because you can see that the energy required to break them here, they are greater much than the thermal energies. The thermal energies when they are available in the room temperatures, 25 degrees or 70 degrees, the human bodies. Here it takes, this point is approximately 0.6 kilocalorie per mole to break in the thermal energies. However, to break the covalent bond, it takes 240 kilocalorie per mole. So that's why covalent bond is quite stable. And in contrast, you can see all the non-covalent interaction here. Non-covalent interactions, they require just only from 1 to 5 kilocalories or per mole and less than the bond energies of the covalent bonds. So who study in the chemistry, I need to remind you about the ionic bonds, or you can search in the Google. You can see that the ionic bond is quite strong bonds, right? But here's we study biology. And in biology, it's not really familiar with you, and I need to put the table here to explain. So you can see that in vacants, the strength of the ionic bond is quite high, right? But in water, those are approximately 30 times weaker, right? So in biology, we have these bond strengths when we put the ionic bond in the weak bonds, because 70% in the living organisms is the waters. So in aqueous environment, ionic bonds are weak bonds. So it's quite a difference in when you study chemistry. And also, however, in biology, when we care about the ionic bond in hydrophobic interiors of the molecules, it's still a strong bond. Please remember it for me. Các vùng kỵ nước ạ. Bên sâu bên trong của phân tử nếu có ionic bond up here thì mình vẫn consider nó là strong bonds, bởi vì here's in vacants ionic bond is strong bond. Okay. You can see here this more is a short video clips about the covalent bonds and also ionic bond.
In today's video, we're looking at how atoms can form covalent bonds by sharing electrons. And we'll also see all different ways that we can draw these covalent bonds. To very quickly recap, we've already seen how some atoms can form ionic bonds by transferring electrons from one atom to the other. This makes oppositely charged ions that are then attracted to each other through electrostatic forces, which bond them together. And the reason that they transfer these electrons is that they both get a full outer shell. Now, this looks great where one of the atoms has too many electrons and the other one has too few, like with sodium and chlorine. But what about when we have two non-metal atoms, like two chlorine atoms? Well, in this case, both of them need an extra electron to get a full outer shell. So keeping electrons to each other isn't going to happen. Instead, what they can do is share some electrons. And each atom needs one extra electron. They each share one
And our short video is about the Arabic sport. And pay attention because some questions in the exam is not only the knowledge from the slide, but also it takes some information from the videos. Come to some source question to revise this part. So, 8 carbon atoms contain 6 protons and 6 neutrons. So, what are its atomic numbers and atomic weight?
Tức là số lượng phân tử và khối lượng.
Now, receiver chemistry in high school.
Yeah.
The atomic numbers is 6, yes, in periodics element we have the atomic numbers. Carbon is 6 and the atomic weight 12, yes. 6 protons plus 6 neutrons. We have the atomic weight, they are 12. And how many, in 6 ways, right? How many electrons do it have?
6.
Yes, 6, the numbers of the electrons are equal to the number of the
Proton.
protons. And next, how many additional electrons will it have to fill the outer shells, and how there is expected carbon's chemical behaviors? 4? 2 or 4? So the first cell covered is 2, and the second cell is 2 electron, and then for the next 4 additional electrons. So how does it affect the carbon chemical behavior? So how they can get stable conditions? So it needs 4 additional electrons to have the full outermost shell electron, so it affects the behavior that they want to share. 4. So the most stable is when it shares 4 additional electrons to other atoms, so they can form 4 covalent bonds with other atoms, right? Other questions. Carbons with atomic weight is 14 is radioactive. So how it differs in structure from non-radioactive carbons, and how does this difference affect the chemical behavior? It's mentioned about the carbon-14 in my slide. So how it differs in structure from the non-radioactive carbons. And how this affects its chemical behavior.
Radioactive carbon have more neutrons than
Yes.
normal carbon, and with that, it tends to disintegrate and probably give out more neutrons than other carbons.
But I want to ask about the chemical behavior. They have 2 more neutrons, so we have in totals molecular weight of 14 instead of 12, right? They have 8 neutrons instead of 6 neutrons to the normal 12 carbons. So how does this affect the the chemical behavior.
I think that it's bigger. I think. What happens?
What happens when you absorb some molecule that comes from the carbon 14s instead of carbon 12s?
I think that it can be radioactive. And because we use the word radio. Oh. Do you have any suggestions? Because I don't really have much knowledge in this.
Chinese. So, okay, so
Just remind me any suggestions.
So, okay, you can see that the chemical behaviors of one atoms is not depend on the number of the neutrons. So it depends on the number of the outer shell electrons. So because the outer shells of the carbon 14s and 12s are the same, so the chemical behaviors of the carbon atoms is identical to the carbon 12s. And only between there are technical problems with to mix with no more energy, so maybe we have break time now. So a little bit sooner.
Alright, so what? We continue the lectures. And we move to the next short questions. It's not really short. Short answer questions, okay. So the element oxygen and sulfur have similar chemical properties because they have both 6 electrons in their outermost electron cells. But in room temperatures, you have the water in liquid form while the hydrogen sulfate in the gas form. So can you explain why might be the case? Yes, please.
Well, actually, the hydrogen bond is just occur with the elements of oxygen, nitrogen, and carbon. So the molecule of H2O has the hydrogen bond, which makes the bond with respect to the molecule is more stable factor, and therefore, in H2O, the room temperature is a little.
Okay, so because the molecules, water molecules, they have the hydrogen bond and they make them more tight, close together, and so they change into the liquid form. So why about the hydrogen sulfate?
Well, for hydrogen sulfate, the hydrogen bond doesn't apply to it.
Why?
Because as far as I know, the hydrogen bond only applies to H2O, NH3, and hydrocarbon.
But my question is why it not appears in the hydrogen sulfate?
The electronegativity,
Yes.
because of the electronegativity of the oxygen, nitrogen, and also the chlorine, the ions.
They are.
They are the top lowest electronegativity. So they can track electrons from the hydrogen.
Lowers electronegativity so they can attract.
The electron from the hydrogen, which can make the hydrogen bond.
What are the sulfur?
Electronegativity of sulfur is too high to apply the hydrogen bond.
If the electronegativity is too high, maybe it's more attractive.
Electronegativity, because at school I studied that the electronegativity is presented with the minus number. So
Think about the size of the nucleus.
the size of the nucleus.
Yes.
So
The sulfur is bigger or smaller compared to the oxygen.
it's bigger, but it's bigger, so the bond with the hydrogen is weaker than compared to
The weaker it's not about the weights of the strands, but we study about the polar. Is less attractive or more attractive?
it's less attractive.
Yeah, because the sulfur have
Sulfur because the electronegativity difference between hydrogen and sulfur is smaller than hydrogen to oxygen.
because they are big, so the outermost electron is less attractive. Is less attracted to the nuclear, the sulfur, so because the latter trace, so it's leading to the is less polar as compared to the hydrogens in the water. Okay, I think I could give you 0.25. So another questions. It's not a big years, but you might wonder why we study more chemistry here. Because we study the chemistry in biology, so you can see that the ionic bond in biology is less strong compared to the ionic bond in chemistry. And also, I'll give you some examples here. You see that we might be toxic by the carbon monoxide. So, journals or who can explain why? Carbon monoxide can be toxic, right? Everyone to give you 0.25, you can use answer what? Please.
So water is really high hydrogenic and it is
Can you speak louder?
yeah. So water can be linked to other molecules because of the hydro bond. Liquid hydro.
Sao?
Hydrogen interact.
Yeah, or I want to ask about is why humans could be toxic by the carbon monoxide? CO instead of CO2.
I know in the biological way that CO can actually block the thiệt là không được ở đây là CO với cả khả năng là ngăn chặn hồng cầu lấy oxy và thải ra CO2 vì thế là nó độc.
How they can prevent these red blood cells takes oxygen and release carbon dioxide? How? By how?
Well, it can due to other. It can.
We study about the chemical bonds. So we can, okay, so here. Explained by the chemical bond way. We studied about the chemical bonds in the first class, so it explains. Can you please? Alright, so my question is why we could be toxic by the carbon monoxide?
If you write down the structure of the carbon
better speak louder, please?
I say that the CO will make the bond with oxygen, will make the bond with oxygen inside the red blood cells. And that leads to that the red blood cells will lack oxygen, which is toxic to our body. You know, that's what I do because I just study biology and stuff. And I just explaining it with my chemistry.
I will give you 0.25 by chemistry, but it's not really in biology. So in biology, in red blood cell, we have one structures or one compound we call hemoglobin, right? Remember? So one hemoglobin can bring for oxygen or carbon dioxide, right? But when with carbons monoxide bind to hemoglobins, you're right because they still are carbon monoxide, they have another two electrons that's unpairs, right? And they can make a covalent bond with hemoglobins. And this kind of covalent bond is not really covalent bond, we call it dative covalent bond or coordinated covalent bonds. Yes, coordinated covalent bonds with hemoglobins. So by that strong bonds, the hemoglobin cannot relieve the carbon monoxide, so there is no space for oxygen, no space for the carbon dioxide, and the hemoglobins cannot transfer any oxygen to our cells, so that's why we make toxic, and somehow the humans they will be dead when they expose a lot of carbon monoxide. So it's 0.25 for use because you can explain the two electrons and about the bond forms inside the red blood cell. Okay. We move to the next part because we don't have a lot of time. So it's about the bonds. Somehow, why we use this chemical bond to explain to so many compounds or many reactions inside biologies. And atoms with chemical bonds can make the molecules, and we move to the first types of molecules inside our body, that is the small molecules in the cells. So you notice we haven't told more. Maybe since now more is 92 elements occurring or naturally in the S curves, and that's among those we have only 25 elements unmixed. Or here's letter S. 25 elements are needed to build the living organisms. And that is the main organisms, remember, C-O-N or C-H-O-N, and they are most frequently found in the cell and forming about 90 or 6% of the human body mass. And I put here the P phosphate and sulfur. P in H here is 99%. Here. C-H-O-N 96, C-H-O-N-P-X 99%. And for the rest, we have a main elements, and for the rest we call the trace elements. So the trace elements are the elements found in a small quantity in the cells, but they are important in the biology's process. So all the compounds in the human bodies, or in the living organism, we can classify into two types. We have the organics or inorganics. Organics is the compound that is, you can find the carbon in them, and inorganic is from the rest. For organic compound, we have a carbohydrate, we have a lipase, we have a protein, and we have also nucleic acids. While inorganics in the living organism is 70% of the living organisms are workers. So all the reactions reaction, Bio's reaction inside the living organism is happened in the aqueous environments because 70% are water. And this is a graph to shows the compositions of the equalized one-type bacteria. You can see that 70% are waters. Nucleic acids is 7%, and among them we have the 1% of DNAs and 6% are RNAs. And our cells are here we have 3 types of ribosomes, 40 types of tRNAs, transfer ribosome, and 1,000 of messenger RNAs. Red place for inorganic ions here occupies only 1%, and for others, mineral trace elements, we have a 0.4%. So it's the first link or to give you the first on the imaginations of the compositions of the elements inside one living organisms. So we move to the inorganic compound, 70% waters. So they are the most abundant substance in the cells and the most intracellular reaction. Intra is inside the cell. So most reaction inside cells is occurring in aqueous environments. And the waters may come from the condensation from the primary atmosphere, release gases from the Earth's interiors, or extra-interior organisms. You don't need to really care about this. Cells are, now we move to the organic compounds, is an important part in the living organisms. There are 4 main types of small organic molecules. We have here sugar, fatty acids, or fat, amino acids, and nucleotides. These are small units, and they can form large units of the cells. We have the polysaccharides, we have the fat, we have the proteins, and we have the nucleic acids. And we move the first part, sugars, or we also have another name that's carbohydrate. For the carbohydrates, they're simply sugar, or we call them monosaccharides. They compose with the general formulas of the CH2O enzymes, where M usually come from 3, 4, 5, or 6. And here is the structure of the glucose monosaccharides, and they can be represented in several ways. So 2 monosaccharides can be linked by colons here. Colon bond, pay attention, here's the colon bonds. And links between 2 monosugars, we have colon bonds named glycosidic bonds. And 2 monosaccharides are linked together by glycosidic bond, and by these ones, by the reactions. Remember the name of the reaction, condensations. In which 2 molecules will join together to form 1 molecule, and with the loss of the water molecules. And in the reverse way, we have the hydrolysis. 反正这个 condensation 的可能。
反正下落。
下落。
啊，我怕他有点。
中了。So. 反正中了。2 molar sugars. So it expels 1 water molecules, and to have the disaccharides. This is covalent bond, pay attention, covalent bonds by condensation reactions. And in reverse, we have hydrolysis. It takes 1 water and separates one molecule into two monosugar. Hydrolysis. Thủy phân đúng không ạ? Chúng ngưng hai đường đơn thành một đường đôi. Thủy phân từ một đường đôi thành hai đường đơn. Và glycosidic bond giữa hai đường đơn và covalent bonds. Very strong, okay? And here the disaccharides, they have formed two monosaccharide molecules and combined together with elements of the molecular waters. And the general form of the disaccharide, we have a C12, 822, and O11 here. Glucose plus glucose, we have a mantose. Glucose fructose, we have a sucrose. And glucose galactose, we have a lactose sugar, okay? They also call they are double sugar, okay?
Đường đôi.
Disaccharide are double sugar. And if so many monosaccharides join together, we have polysaccharide đường đa. So they are polymers of the simple sugars of the monosaccharides. Many monosaccharides join together with reactions. Yes, condensation reaction. So remove so many water molecules. We have the polysaccharides. And there are two forms of the polysaccharides. The homogeneous polymers, they contain only one kind of sugars. And because this is only one kind of sugar, we consider them as non-informationers. And there's the other way, we have the heterogeneous polymers. They contain from 8 to 2 types of sugar. So the type of sugar in human and animal, we have the glycosins. And in plant, we have a starch and the cellulose. So what are the functions of the polysaccharides? The most important function of the carbohydrate, can you guess?
Chức năng của polysaccharide.
Carbohydrate in general.
Chức năng của provide energy.
Yes, provide energies. They are the energy sources that the most important functions of the carbohydrates. So here we have the glucose, they have the key energy source for the cells, and in the series reaction is broken down in small molecule and release the energies, and the cells can use the simple polysaccharides. And here this series reaction here in eukaryotes, they happen in mitochondria. So respiration inside our cells here. They use glucose, they break them down into small molecules, and by this type of reaction, the energy is relieved. And the next function is a very important function of carbohydrates, and their structure from support. You have it in the cellulose form in the plant cells. They are the main components of the cell walls. And the cellulose, they are the most abundant organic chemical on Earth. And around the chitins are the linear polymers and acetylglucosines, and insect exoskeleton, and also the fungus cell wall. So when you burn the insects, whenever you burn the insect, you can feel one type of the same, I think the smell of chitin when you burn it, you can see the same kind of smell, because the exoskeleton of the insect chitin You can try it at home.
We catch insect and then we burn it fire. We can smell the chutney.
And the other function, the last functions of the carbohydrates, we have the molecular markers. When the carbohydrates connect with the proteins or when they connect with the lipids, we have the glycoproteins and we have the glycolipids, the cell membranes. So it's where selective recognition by the other cells on the healthy adherent to others on the blood time are generating importance. The human blood time is defined by the glycoproteins in weight, you know. The human blood time. In which cell do you know? The O blood type, AB blood type, A or B difference because they have the different glycoprotein in which cell? Nào, mình có các nhóm máu khác nhau bởi vì trên tế bào nào có các glycoprotein khác nhau nhỉ? Yes, in red blood cells, because carbohydrates have the very important in the molecular marker. Here. So next we go to discover another organic compound inside cell. This is lipid. The lipids come from a Greek word. Lipos means fat, and they are non-polar. They are non-polars, so that's why they are not soluble in water. And they soluble in non-polar solvents, such as chloroforms. So when we want to isolate some things, have the lipids form, we solve it in the chloroform. And they have a different two structural parts. We have a hydrophilic head here. We have a hydrophobic head, the chemical active, and we have the hydrophobic tails, and the difference between the hydrocarbon trains and the not very active cathode. Hydrophilic nước, hydrophobic kỵ nước. So the properties of the fats depends on the length and the saturation and fatty acid train they carries, and this is one very important lipids. This is the phospholipid. Phospholipids, the structure of the phospholipid molecules, they are composed of the two hydrophobic fatty acid tails and joint or hydrophilic head. So the hydrophobic tails pack together to exclude the water and forming lipid bilayers here. And these layers is actually our cell membrane layer. cell membrane is made of phospholipids. Who can explain me, so why is here we have a saturated fatty acids and unsaturated fatty acids? What difference between them? Yes, please.
The saturated fatty acids are only combined with single bonds, and the unsaturated have double bonds or triple bonds between the topics.
So unsaturated fatty acids contain the double bond of carbon. Double bond between carbon atoms. Saturated is no, so that's why you can see the shape of the saturated and unsaturated like this. So what about the characteristics of the unsaturated fatty acids? In room temperatures and liquids, liquid form.
Well, the saturated fatty acid in room temperature can be a solid form, but however, the unsaturated one can be a liquid form. Yeah, so when applied into the phospholipid bilayer, I think it can bend the unsaturated head so that it can create a hole inside of the membrane.
And about the fat in the human bodies, can you get that this contains more unsaturated fatty acids or more saturated fatty acids?
In the more saturated one, and because it is unsaturated, so it is more flexible, and that can adapt more inside of the cell environment.
More definitely.
And it's more
Bọn này mỗi lần trả lời câu hỏi có các tổ tư vấn. So we move to the next line about the function of the lipids. The first very important function, they are energy source. So they store in the cytoplasm many cells in the form of droplet of the three tricyclic molecules. So they can produce about 6 times much usable energies weight for weight as glucose. And in animals we have a fat. And the fat contains only saturated fatty acids. And in plant we have oil, corn or olive oil. So the oils contains unsaturated fatty acids. So the fats and oils, 6 times, this is why 6 times. The fats and oils provides maybe 38 kilojoule per kilogram. While glucose is only 18, I think. So that's for the energy source. Lipids provides more energies compared to the glucose. But in the cells, lipids more in the form, the storage form, the storage energy source. But the sugars, they easily to break down sugar into smaller parts to really energy. So we can call the active source of energies is the sugars, but the storage energy source, we have the lipid. And the next very important functions of the lipid, they are the contraction of biology membranes. Like the most important chemicals in the phospholipids in the previous slide I showed you, and also we have glycolipids in these in the surface of the membranes, and also the cholesterol. They support the biomembranes. So we, by some advertising and by some research, the humans also fear the cholesterol, but cholesterol is really important in the structure functions inside cells. Without cholesterol, is maybe you will make a big problem when you lack a cholesterol. So we move to the next compound, very important compound. Is it protein? So the protein, these basic units of proteins, we have the amino acids. So this is the amino acids all have the carboxylic acid group and amino group. Here's carboxylic group, one amino group, and connected with the alpha carbons, and also one side chain. So at the pH are 7, both the amino acids and carboxylic group are ionized. Here, in pH 7, they are ionized. So what makes the amino acid different? The amino group are the same, right? The carboxyl group, the same. Alpha carbons, the same also. So the difference of the amino acids, they only difference in their side chains. Here, the side chain, you have the side chain R. So RS commonly is one of 20 different side chains. Here we have 20 different amino acids. We have 20 different side chains, and SPS 7 amino acids and carboxyl group are ionized. So two amino acids can combine together. The peptide binds against with reactions, condensation reactions. So the condensation between the carboxyl group of one and the other amino group of the others, and they result a bond linked to amino acids. We call them the peptide bond. Giờ mình học là peptide bond nữa. Đúng rồi, lipid peptides. And they are weak ties are born here. Weak bonds are covalent bonds. Here's also strong bonds, and here's also the covalent bonds again. And by the reverse reaction here, two amino acids linked by the peptide bonds can separate into two separated amino acids by hydrolysis against the same. Okay? Remember. And so you know that we have roughly 200 amino acids identified in nature, but we have only 20 amino acids found in living organisms. Do you know the reason why we have only 12?
We arrange differently in every different type of protein.
Yes, it can arrange differently, make different type of proteins. Why 19 or why 21, why 30s, why 50s, and why 10s? Why only have 20 amino acids composed by 20 different side chains? Our side chain is enough. Think about example case, and now it's in among 20 amino acids found in the living organism. I introduce here that's 3 special amino acids. We have the cysteine, glycine, and proline. So why are they important? So the cysteines contain the S8 group, and they can form the common bond with other cysteines. So you can get weak ties of protein that we can find so many ties of the cysteines. In protein, that is, we want them more strong or stable. They must contain more cysteines because they can form the common bonds with the other cysteines in that protein. And also, when they can form different cysteines, they can fold that kind of protein. You know, maybe the first cysteine in the number 3 and the next cysteine in the number 12, when they form the disulfide, they can fold that proteins, right? And make these more stable, it's more strong. At the same time, the proteins we have proline. The proline, they have the R group that link back to its own amino acid group. They're forming a ring structures. And this method is similar to the typical structure of the amino acids, and they have no longer a standard amino group. They have no NH3, so it makes these kinds of amino acids very rigid. And the last one is the smallest amino acids, you have the glycines, and a single hydrogen atom. As its R group, R group is very simple. Only 1 hydrogen. So it's more size, so it's allowed to fit in to the tiny space, so that the rows of these glycine. So that is 3 special amino acids that I have introduced to you. The next is the hierarchical structure of the proteins. And we have how many levels structure of protein? How many levels? It has 4. Yes, we have 4 levels. The first level is primary structures. Primary structure is just a chain of amino acids. Is a primary level structures. The secondary structures is the folding polypeptide trait amyloidase into alpha helix or beta sheet. Các bạn nhớ trong high school mình có gì nhỉ? Cấu trúc bậc hai protein là xoắn beta hoặc là gốc mẹ này, xoắn alpha hay gốc nếp beta. And the tertiary structures. In the structures, secondary structures with the fibers move and turn into single polypeptide chain packed into the larger intervannar straight or table structures. So there's so many sections, so many parts of the proteins with the alpha helix or beta sheets packed together. We have the tertiary structure. And for the last, quaternary structures. For quaternary structures, just imagine that we have two or three, more than two tertiary structures staying together, so we have the quaternary structures. So we have multi-chain complexes. That is for structure of the proteins, and we have the next part is denaturation and the folding proteins. When a protein loses its highest order structures, but not its primary sequence, is said to be denatured. And the factors may affect the proteins. We have the temperatures, and we have also the pH. And the denatured proteins are usually non-functionals. So some proteins can reverse after denaturation, even when they are alone in the test tube, but it's not a lot. So here is one example of our denatured protein. You know the white part of the eggs? They're in this liquid form, but when we heat them, it changes into the solid form because the albumin. We have our albumin, or in these white parts of the eggs, we have a lot of protein called the albumin. And when we provide a heat to the eggs, the albumin will be saturated and it changes into the solid form. So what is the roles of the proteins? Proteins inside our bodies have so many types of functions. First, they have the regulation function. They can control all the activities. They control activity inside cells. And also our structures. So many types of proteins can involve in the structure function. Like we have tubulins or microtubulars is the main components of the cytoskeletons, the skeleton of the cells, and histone are in the chromosomes. It is one proteins involved in the structure functions. And so many types other functions. We have a signaling. We have a transport. This can bring small molecules and ion across membranes, catalyze on enzymes. motor protein that generates forces for the movement of the vesicles of the molecules inside the cell. We have a movement. And very important are proteins compound, it is the enzymes. Our enzyme is most of the kinds of the enzyme, so mostly the majorities of enzyme is composed from proteins. So the enzyme helps speed up the metabolisms or chemical reaction inside our bodies, and they do not change at the end of the reaction. This is the enzymes. And do you know the name of the enzymes? How we can name one enzyme? We have a suffix, ase, remember? Name of one enzymes. Subtrace plus ase. So it is named by taking its subtrace and we add the suffix ase. For example, we have protease is the enzyme that catalyzes proteins, right? So protease is catalyzed proteins, and this is named is do not apply to the enzyme discovers before these rules are introduced. So we have a, for example, we have a trypsin, right? We have trypsins, we have a ranings for so many types of enzyme that is not in that rules. And the characteristic of the enzymes with the small amounts needed to catalyze a lot of substrates, and each class of enzyme will catalyze only one particular reaction. The enzyme catalyze reversible reaction, and many enzymes only work with present of the coenzymes or cofactors, which may be sometimes maybe organic compounds like we have or magnesium or zinc or so many types of inorganic compounds can work with the presence of the enzymes. And the enzymes basically they are proteins, so they can be affected by change in the temperatures and the pH also. Because they are proteins, but most of the enzyme is protein, but small number of enzymes are not proteins, but they consist are the RNA molecules. So the mechanisms action of the enzyme activities, they have is molecule have a very precise shape called the active site. The active site. And the substrate here is fit into the active site of the enzymes like a key and the lock, right? So here we have the key and the lock here, and here the key. So the reaction will take place at the active sites. We have the active site here. They take place at the active site to form a product, and the products have a different shape. From the substrate and therefore repel from the active site in one step, while the substrate fits into the active site. The step 2, the forming of the enzyme subjects complex and reaction take place into an active site and the test. Step number 3, the products repels from the active sites. So there's so many factors could affect the activity of enzymes. We have the first, the pH. So each enzyme have the optimum pH or as weak its rate to reaction fastest. You have the, for example, the amylase pH 7, like trypsin pH 8 to 9, or pepsin in the pepsin in our stomach is work at the pH maybe around 2, right? So the next factor is like the activity of the enzyme. We have the temperatures. So the rate of reaction increase up to maximum in we call the optimum temperatures, and after optimum temperatures, maybe in our human body is around 70s or 37 degrees, and the rate of reaction falls quickly because the bond main structure of the enzyme start to break, and the active site will move itself, again because of protein denaturation, right? So if maybe it's 60 degrees, the enzyme activity will stop together because the enzyme is denaturate. So I emphasize this part because in last week there's one student asked me why we need to use the Cas polymerase from archaeas. Why we cannot use the human enzyme to perform the PCR reaction. The PCR reaction have the one step that's for the DNA polymerase work is 60 to or 65, 50 to 65 degrees. So at that temperature, the human enzyme cannot work. It's because most of the human enzymes is made of protein, and at that our temperatures 55 to 65, the human enzyme will be denaturate. They lock the active site. The enzyme will be breaks, and the active site lose their safe, and there's no more work. So what about the Cas polymerase is still, the Cas is still proteins, but by different way of foldings, they can work. They do not saturate or denaturate at that times or temperatures. And it's all because of evolutions. So we can live and work normally at temperatures around 37 because of evolutionary. That's why we have ears, and for the archaeas, they live in the very hot temperatures. So the protein of them will be need to fold so many times to reduce the entrophies, folding penalties, and they can resist the high temperatures like this. So when we got fevers, why we need to stop or why we slow down our bodies quickly? Because if our temperatures is more than 40, or for so long, we will get very tired because so many types of enzymes inside our bodies started to lose their activities, and all the reaction inside our bodies are starting to slow down, right? So we need to, when we get fevers for a very long time, we take some pills to slow down our temperatures. And for the next factor affect the activity of enzymes, we have the concentration of the enzymes. So, if we increase the concentration of the enzyme, there is more chances of the enzyme in the substrates or collisions, and the rate of reaction. So, if the concentration of the enzyme increase, the reactions increase. And the next, the concentration of the substrates is more the same. When we increase the number of the substrates, is more chances of the enzyme and substrates mixed together. But if it's too much, the reaction rate will be decreased with too much substrates. We don't have enough enzymes. Okay. So questions for this part, for the protein part. Can we synthesize and inject protein into people without the body resisting synthesized proteins? So in the previous case, we need to use the enzymes or this meta protein from others organisms to perform the PCR reactions in the human genes. So in the reverse case, can we synthesize and inject proteins into the fibers that cannot produce that kind of protein and inject that kind of protein inside them? We use the proteins from bacteria, or we use the enzymes based protein from bacteria to multiply the human gene in the PCR. So can we produce the protein that can be injected in our bodies by the other source? So can we produce the human proteins and inject in our bodies? So you know that when we inject a protein that's not human proteins inside our bodies will be denied them. right? And so many reaction. Oh, okay, this is please speak louder. Okay, your idea that we can synthesize proteins even it's not humans, and then we added the glycoprotein inside them. Glycoprotein and glycolipids. But the glycoprotein and glycolipids is appear in the surface of the cell membrane only. So maybe your idea is that you need to produce protein A's, and then we added the glycoprotein or glycolipids in that protein A's over the surface. Now your protein is the side chains of the amino acids only. When could you add it? The glycoprotein or glycolipids in this? Is it easy? Yes, quite hard. And it's not your protein anymore when you add it sometime like this. It's not your protein. Okay. Or think about the diabetes patients. I agree with you. We can synthesize and inject proteins again. We can use the enzyme for bacteria to perform or to multiply the human genes in the PCR reactions. And also, in the reverse case, we can use the bacteria to synthesize the human proteins and then to inject it back into the human bodies. So think about the diabetes patients when it's like or they really need to provide the insulin. This one kind of enzymes in the human body is really important in glucose, metabolize the glucose, right? So the diabetic patient, they need to inject the insulin, and in the pot, or there is not, or we cannot find the insulin stored or in nature. So we just kill the cow, and we isolate the insulin from the cow, and then inject into the human to treat or to help the humans with the diabetes. But it's quite expensive, it is quite difficult, and there is a lot of contaminations when you purify the insulin from the cow. Maybe there's so many other types of proteins and other compounds will be contaminated in that kind of insulin. So it makes the diabetes patient get dangerous. Even that kind of insulin is quite expansive. And then we can take the gene from the humans. We insert that kind of genes into the plasmid of the bacteria, and we use bacteria to synthesize the insulins of humans, and then we purify our own insulins. It's cheap and it's more safe, so it can apply in the large population of the patient, and it costs a lot of the diabetes patient. Okay, 0.25 for use. We move to the next part about the nucleic acids. So the nucleic acids is the basic units of nucleic acids. We have the nucleotide. So what are the two nucleic acids inside our bodies? Remember? Nucleic có hai loại đâu? Nucleic acid.
Nucleic acid ở trong cơ thể ạ?
Yes. We have DNA and RNA. Yes, two types of nucleic acids. And the basic units of the nucleic acids, we have nucleotide. The nucleotide is made of three parts. First, phosphate group. Second, sugar. And last is the base. So phosphate group at the same, but the sugars, here is pentose sugars. We have two types of sugars. We have the ribonucleic acids by ribose. This ribose sugars is used in ribonucleic acids, it's in RNAs. And we have the deoxyribonucleic nucleic acid, we have a DNA. So DNA and RNA different in the types of sugars and also different in the base. The first part of the base, we have two types of bases. Pyrimidines delete from pyrimidine rings. We have a cytosine, thymine, and uracil. Uracil is only appear in RNAs. So I remember in the finance exam in next year, I don't remember, but there is a question asked about the genetic materials of a virus. So whenever it is appear, the uracil is mean the genetic material as that virus are RNAs. And the next group of base, we have the purines. They have the 5 membrane rings fused to the 6 membrane rings. They are bigger, 6 membrane rings, and we have adenine and guanine. So in total, we have 5 bases. Uracil only appears in RNAs. Pyrimidine is smaller base, and purines are bigger base. So the nucleus or nucleotide is named according to its nitrogen base. And the most important roles of nucleic acids is they are storing the biological functions. 2 types of nucleic acids in our bodies, RNAs and DNAs. RNA and DNA different in the type of sugars. Ribonucleic sugars is the RNAs and deoxyribose, we have the DNAs. And also in RNAs we have uracil instead of thymines here. So in Vietnamese, when you study biology in Vietnamese, we have A, T, G, and X, right? That means the 16 in Vietnamese. Nếu các bạn học cấp 3, các bạn học môn di truyền học ạ, mình sẽ có 4 nu là ATGX đúng không? Cái X đấy vẫn là 16 nhưng mà được các dịch giả dịch ra tiếng Việt rồi. Còn cái C ở đây trong tiếng Anh thì nó vẫn là 16, but in English word, okay? So now we study genetics, or we study our DNA. A, T, G, and C. It's not X anymore. But it's the same X like you study in your high school. So here the DNA and its building blocks. We have a double strand of DNAs, Y, C, complementary by 3 hydrogen bonds right here. C, complementary with 3 hydrogen bonds with one. Two and 3 hydrogen bonds formed between guanine and cytosines, and only 2 hydrogen bonds formed between adenine and thymine. So when one gene is one important gene, or one gene is one make them more stable, it means the GC content in that gene is high or low. So if one gene is one bit more conserved, this could increase or decrease the level of GC. So okay, it's because C and G is complementary by 3 hydrogen bonds. So if you want to make your DNA or you want to make your gene is more stable, is more strong, this means you need to increase the levels of the GC content inside your cell, inside your gene, okay? Next is the code information DNA, is how this is converted into amino acid sequence proteins by multiple steps. And we have here's one, the first DNAs will be transcription factor bind to the DNAs and the regulatory regions and controls and activate them. And step 2, multiples are initial complex bound to the DNAs, and RNA polymerase started to work, and this is the transcription process. We have a pre-RNAs, and then we can cut off part of the pre-mRNAs. This is the part we call non-coding region, or the intron. non-coordinating intron here. The coordinating region called the axon. So when we cut off the introns, we have the mature mRNAs, and then the mRNAs will be translated into the proteins here. So how we can translate the genetic information into how we can read it, translate them into the protein that defines who are we, okay? Is such a question, but we'll come back later. We have also the one videos about the biological molecules, and maybe you can see it at home. We have here the next part, the molecule complementary. So the molecule complementaries of the two proteins are, let's permit them bind more tightly than the two non-complementary proteins. You can see that the stable complex between the protein A and B, like they can form the ionic bond together, they can form the hydrogen bonds, in which part they have the hydrophobic interaction, van der Waals interactions, so this makes two proteins more stable. Whereas with protein A and C, you see that there is less weak bond formed between them. So the molecule complementaries is the keys and the locks fit between the host shape, chains, and other physical properties and complementary. And the multiple non-covalent interaction form between complementary molecules cause them bind tightly. Intramolecule complementary establish the 3D shape of protein. So why we studies about complementary, you can see that for the subunit here, the basic units, you have basic units, molecules inside our cells. We have the sugar, that is the monomers of carbohydrate or polysaccharides. Amino acids is the basic units of proteins. Nucleotides, the basic units are nucleic acids. And by covalent bonds, the first step, they need to bind together by covalent bonds. By which reaction, remember? Condensation reaction, covalent bonds. They can make the macromolecule. So sugars have the polysaccharides, amino acids, protein, nucleic acids, nucleotide makes nucleic acids. So here's one example. It's from the amino acids make the global protein, and nucleotide make the RNAs by covalent bonds. And by non-covalent bonds here, by non-covalent interactions, they can make the macromolecular assemblies. And here from the protein and RNA, they can make structures inside a cell is the ribosome. Okay, covalent bond make macromolecules, non-covalent bond make the macromolecules assemblies. And together, covalent bonds and non-covalent bonds have all the things in our bodies work smoothly, work together. So that's the end of the lecture today, and we can come back about some short answer questions in the DNA part, okay? So with the following statement, correct and explain your answers. A DNA strand has polarities because its two ends contain different bases. No answers? No one has the answer? Okay, I think you're too tired, so we can finish the lectures now.